With that, I'm going to turn the stage over to Evan Hecker. Evan, as most of you know, as I mentioned, is the husband of Kara. Also father of Nathan, who is battling a PLGA. What you may not have known about Evan is that he has worked in drug discovery for the last decade, and he was after 2 years of drug discovery work in industry, and then he went back for his Ph.D. He's a lot smarter than he looks. In organic synthesis from UT Boston. You're going to get me back, aren't you?
They know it's true. They've seen my face.
He also completed his postdoctoral fellowship at Harvard, and for the last 6.5 or 7 years, he's had experience in antibiotics and oncology drug development. He is soon to be the director of API development at Spero Therapeutics. Evan is going to take us through the the progress of science for PLGA. So let's welcome Evan.
It doesn't matter. I'm smart. Okay, so what I thought I would do is, because there's a lot of families that we've known for the last few years, I thought I would just briefly tell you my story and how I got involved with the foundation, just briefly, because I only have a few minutes. And I think it should maybe tell you a little bit about where I'm coming from in this. So, Nathan was diagnosed in 2012, and I was in the hospital with him. He was getting surgery, and a longtime friend of mine came to visit me, and we were sitting there, and you know, we've all been there. You're devastated watching your child go through this, especially before you don't really know much. And my friend looks at me and he says, "You have to ride the PMC." And I said, "I don't know what that is." And he said, "It's a 165-mile bike ride." I said, "It's not going to happen." And he said, "No, you're going to do it." And I said, "Shut up." And he said, "No, you're going to do it." I said, "I don't have a bike." And he said, "Well, we'll figure that out." So anyway, I bought a bike the next year, and I rode with him, and you know, it was more just about trying to take back some control over what is an uncontrollable situation. And then the first year, we only raised about $11,000, and that was a tremendous amount of money for me and my friend. But the point is, I say only because it started something, and the next year there was 5 of us, the next year there was 9, this year there's 13. Every year we raised a little bit more, and the point is just to follow on Amy's point. You know, it can be really daunting to say, hey, I can't go raise $50,000 or $100,000, and that's not the point. Especially there are some local events where it's more important for you to just be there with your families and raise whatever you can raise, and just be there and experience it and see the importance, and then share that with other friends, because who knows, you may have a friend who says, wow, that's really incredible, I want to help out, how can I help? So, you know, do it when you're ready. And to follow on to that point, Nathan was diagnosed in 2012. His first stable MRI after chemo was in 2014. And it took us a while to get through all of his PT and OT issues, and we're still dealing with behavioral stuff that we'll talk about later today. But the point is that it wasn't until, and on that first PMC ride, I had met John. We were just riding, and I saw his jersey, and I said, wow, what's PLGA Foundation? We struck up a conversation. But it really took about 1.5 years for me to have sort of the clarity of mind to sort of be beyond the crisis mode dealing with Nathan, that I was able to reach out to John and Amy and everybody and say, hey, what can I do to help more? So the other thing I would just say before I really get into it is, do what you can when you can. And don't feel pressure from, if you're dealing with your child right now, then deal with your child. That's the most important thing. If you can help. Okay, so what I wanted to talk briefly about was, I think the way I would describe this is, how is the low-grade glioma world changing, and why is it still the same? In other words, there's a lot of progress. We always hear about all the research going on, and yet for us, it feels like it's still the same. You know, just to follow on specifically what I mean, a couple of things that I heard yesterday rang true for most of the families. child had some issue, they brought you to the doctor, they did surgery, they biopsied it. They looked at the pathology and they diagnosed it as an oligodendroglioma or ganglioma or a pilocytic astrocytoma, and you did carbon crystal. And then if you failed on that, you went to TPCV. And that is a pretty common theme. And the other theme was there was growth, there was progression, and we started in chemo. So that, to me, that breaks down into three things that I saw a lot of progress on at ISPNO, which I'll get to in a second. But really that breaks down into three things. The diagnosis. You're in the clinic, you get a biopsy, and they do a pathology. That's how you're diagnosed. Then you're treated with carbon-crystine, that's the front line. And progression is usually defined as growth. And that what I would say is an outcome. So really, when I was at ISPNO, what I was really struck by is the breadth of research in both diagnoses and treatment, but as well as outcomes. And I'm just going to run through briefly kind of where the field is going and hopefully how the world will be different for our kids based on what's happening at ISPNO. So ISPNO is the International Symposium on Pediatric Neuro-Oncology. It is a biannual, I wrote it down in exact words, a major biannual global meeting with blah, 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 blah. It is the premier event for researchers in pediatric neuro-oncology in the world. There are researchers from all over the world, and they gather every other year in an exotic locale like Liverpool, England, to share their research and to a certain extent come to consensus about the next steps for research and what that treatment looks like. So again, I'm going to run through these kind of individually. This was a slide, and just to be clear, I'm not presenting the research per se. I'm presenting a kind of a high level, what is happening in the field and what can we expect to see hopefully in the future. So I was struck by this. This was a slide that somebody presented on the state of affairs in pediatric neuro-oncology treatment. And in the 1930s, it was primarily surgery. They had nothing else. And then in the 70s, radiation came on and really, I can adjust this. Everybody can hear me, right?
Yeah.
In the 1970s, it was radiation. There were a lot of different techniques for radiation. And in the 1990s, Roger Packer, that's not what I was going to do. In the 1990s, Roger Packer pioneered the crystalline carbon protocol. That is famously called, I believe it's 9925, which refers to the fact that it was done in the 90s. It was a clinical trial. So this is standard of care. TPCV and Blastine are also alternative treatments that many of our kids have been on. But really, these are not, I don't know what I'm doing. These are not targeted therapies. These are just chemotherapies, which means they just kill cells. And we hope that they kill more cancer cells than normal cells. The next stage is to look at what are called targeted therapies, which hopefully will target only the cancer cells. And that is really the cutting edge of where the field is. Okay, so diagnoses. One of the big things that has come out of research in the last few years has been this idea of a lot of astrocytomas and low-grade gliomas being what's called a single pathway disease. And really what that means is that there are a lot of fundamental proteins, enzymes, genes, targets that people can attack with drugs that are all in the same cascade of biological things that happen in cells, such that there are a lot of different ways we can attack these low-grade gliomas that are all very similar. And so what that means is that you can look at a lot of the different genetic mutations that are present in some of these tumors as maybe a broader context of a way to approach treating the disease. And what that means is that they can fold in genetic profiling as a diagnostic tool. One of the things I heard several researchers say is in the future there may not be terms such as pilocytic astrocytoma, oligodendroglioma. You'll be diagnosed based on the genetic profile of the tumor. In other words, a BRAF fusion equals a pilocytic astrocytoma most of the time, regardless of where it is. They'll just refer to it as a BRAF fusion tumor instead of a pilocytic astrocytoma. And I'll get to why that's important in a few minutes. So, a lot of what's happened in the field in terms of targeted therapies has been focused around BRAF historically. And that's because BRAF is a target that has been seen to be present in a lot of different cancers. And so drug companies develop drugs for BRAF, lots of different drugs for BRAF. And we, as a community, focused on pediatric cancers, could co-opt those existing drugs and see if they're good for our kids if they have BRAF. But for a lot of our kids, they don't have BRAF. So what does that mean? And so what this slide shows is basically a huge number of different targets that are present and even sub-targets. So for example, this orange line is BRAF fusion. So that is what many people know as the BRAF, KIA, 1549 fusion protein. The point is that's different from the BRAF V600E. And so you're able to break this down so that of all of the histologies being, say, pilocytic astrocytoma, polymyxoid astrocytoma, oligodendroglioma, you can then look at which of these have BRAF fusion. Some of them have FGFR1, MIB, different kinds of MIBs, IDH1, etc. And then you can take that information and look at which drugs may be good for those. So the point is that hopefully in the future, they'll be able to look at this kind of information as a diagnostic tool. And they're starting to do those things. The other thing that I saw a lot of is people looking at the locations of tumors and correlating that to not only outcomes with the different types of drugs and the different genetic profile. So in the future, what you can imagine is a child comes in, and after they do a small biopsy, they know the genetic profile of the tumor. And they can couple that with the location of the tumor, depending on where it is in the brain, and they'll know that most of the time those tumors have BRAF V600E or fusion or whatever. And they can use that with the genetics to come up with a very specific diagnosis. And why is this important? One of the things I saw there was a presentation of medulloblastoma, and basically for kids nowadays with medulloblastoma, if a child comes in with medulloblastoma, the new way they'll be diagnosed is coupling with the genetic profile. And they're broken down into 3 subtypes. And then further broken down based on risk categories, such that if a child comes in with a medulloblastoma, they can basically be pinpointed for the treatment that they will do best on. So it's not just a blanket, they have a brain tumor, let's give them carbamazepine. It's that they have a medulloblastoma with the Wnt pathway, and therefore we know it could be this type of treatment that's going to be most effective. And that is really where I think the field is going for our kids. So in the future we'll be able to say, your child comes in, and they're diagnosed with BRAF fusion protein, and we know that it's going to respond best to this type of drug first. But for now, carbamazepine is still the front line, and TPCB is still the second line. And there are lots of targeted therapies for BRAF V600E, but at the end of the day, they're not quite ready for the broader, as you saw, a lot of the different genetic profiles. So what else is there? So beyond BRAF V600E, which is where all the drug companies have been focused for a lot of years, there are now a lot of drugs, so-called MEK inhibitors. So MEK is just farther down that single pathway, which means the way the drugs work for BRAF V600E is very specific for BRAF V600E. But these MEK inhibitors theoretically would do the same thing for all the BRAF V600E as well as all of our other genetic mutations. So there are a lot of different MEK inhibitors, a lot of companies have been very interested in it. And fundamentally, there's trametinib and cobimetinib, which are both approved drugs, but not for our kids. And then there's selumetinib and binimetinib, both in phase 2 development by drug companies. And some of our kids may be on some of these. Trametinib and selumetinib are the 2 that I think are probably the most heavily investigated for our kids. There's also MEK162 and TAC580. What happens generally in drug development is a chemist will make a drug and they'll give it a number based on some company's numbering system, and they don't know if it's any good. They make thousands, millions of drugs. All of a sudden, they think it might be good. They give it a funny name like this that ends in some weird thing that's consistent with everything. So you can tell basically that these are much earlier than these because they don't have that funny name. So the take-home message here is that these are all farther along than these, but yet these might actually be the more promising for some of our kids. And some of the research at Dana-Farber is looking at specifically TAC580. And there's a clinical trial that we'll talk about in a moment that's looking at MEK162 in LA. And so, again, there's going to be stuff that's beyond the MEK inhibitors, but this is BUF V600E was something we heard a lot about in the last couple of years at Camp Central from Adam Resnick. And MEK inhibitors are going to be the thing that we start to hear more and more about. And there's going to be other things beyond that. So clinical trials on the horizon, this is probably what you're most interested in terms of what will have the most immediate impact to kids that are going to progress. And there are 3 drugs that I'll just talk about, or 3 clinical trials. The first is this LOGIC trial. This is a trial that's going on in Europe that the foundation is very active in funding. And specifically what this trial is looking at, and this is, in my opinion, a really, really important trial that is going to have a huge impact on our kids. This is a direct comparison of carbovincristine, vinblastine, and one of those MEK inhibitors. And what they're going to do is a child will come in who's never been treated with the drug, and they'll biopsy the tumor, and they'll have some genetic information. They'll know whether they have BRAF or fusion or IDH1 or whatever other genetic abnormality that's in the tumor tissue. And they'll randomize them, so some of those kids will go on carbon, some will go on vinblastine, some will go on the MEK inhibitor. And then they'll look at where the kids do better. And so hopefully, there's going to be a clear differentiation so that you'll see certain kids with certain genetic mutations do better on carbon, and certain ones do better on a MEK inhibitor. But what this really means is that this trial is directly comparing one of these new drugs to the current standard of care. So what that means is that in the future, a child will come in and not necessarily be given Carbovic or TPCD because they have nothing else to give them. So this is a really important trial that we're getting underway, and specifically, the foundation is funding, in my opinion, one of the most important parts of this, which is the genetic profiling. So without the money that the foundation has raised, this trial may be happening, but without this genetic information. And that would be, that would not help our kids as much as it's going to help. So this is a really landmark opportunity for us. On a side note, we got to meet with Stephen Pfister at ISPNO, who's the lead researcher on this trial, and he's handling a lot of molecular profiling. And I was struck by how passionate he was just for our kids. So just to follow on Amy's point about the foundation is not a hub of Dana-Farber, we're funding a trial in Europe that probably very few of our kids in the US will have access to, but will have a broad impact to the future for our kids. And funding researchers across the world.
How many kids are going to be in that trial?
So I don't think the number is firmly set yet. So it's going to be in 23 countries. Maybe Amy knows the answer.
Yeah. So the that trial is 23 countries are going to be involved, and they are estimating 3,000 kids. It is the largest trial ever. Wow.
And one of the things that's really cool about this too is this is going to be adaptive, which means they're going to be getting real-time data. Usually when you run a clinical trial, you treat 1,000 people and you don't know if the drug works until after you've done 1,000 kids. They're going to be getting data along the way, so that if early on, if they see that certain kids with a certain mutation are really thriving on this MEK inhibitor or carbovincristine or vice versa, they can adapt the trial to take a closer look at that.
That kind of leads into my next question, which is if this trial shows that these kids with this genetic profile are doing really well on the clinical trial drug, do the kids in the control groups get an opportunity to get treated with that later on,
so
if they need it?
It depends. So if the children are doing well in the drug they're on,
and they don't need to get treated again.
Change.
Right.
If the kids progress, and I'll talk about what that means in a minute, if the kids progress, then they would probably, that would be an endpoint to the trial,
Okay.
and they would probably not be on the trial anymore. There are ways to keep them on, it's called compassionate use, and then depending if they have an adaptive, in the start probably not, but if they start to see a reason that if one does really well over the other, they'll be able to go to the regulatory agencies to see if they want to try it out.
Evan, can you clarify why the LOGIC trial is different from what's happening in the U.S.?
Sure.
And what's important about this? Because I know that there are MEK trials also going on in the U.S. So what's different about what's happening in Heidelberg?
So the MEK trials in the U.S., there's a segmented trial, I believe it's in St. Jude's, and then there's a MEK 162 that's getting started up in CHLA. And then there's the TAC 580 that is going to be starting at Dana-Farber. But those trials, in fact, all the trials in the U.S., every single one in the U.S., in order to be on that trial, your child must have failed off carbamazepine or TPCV, or something like that. That would be a requirement to be on the trial. So what that means is that even if the drug looks really amazing, all the data says is that that drug works for kids that have failed on carbamazepine and TPCV. So that in the future, that data will say kids who've been on TPCV may benefit from this, but it won't change the fact that kids will still come in and probably get carbamazepine and TPCV. So the trial in Europe is the first one that's doing head-to-head. So that has the potential to change that paradigm. That has the potential to say, cellular or whatever the mechanism is, works better than carbamazepine. And there's no reason to treat certain kids with carbamazepine.
It's fantastic.
What's the endpoints of the trial? Because stability is historically not an oncology endpoint.
Yeah, so I think it's progression-free survival.
Can you repeat the question?
What is the endpoint of the trial? So when you do a trial, it's not just do the kids do well, you have like a clinical endpoint, they're evaluated. There's going to be some other endpoints, things like if the visual acuity decreases and other things like that. To be completely fair, the protocol hasn't been finalized yet, so I think some of that is a negotiation among the different countries. And one of the big things they're waiting on is the fact that selumetinib has some phase 2 data that's going to come out soon. I think they're hopeful that selumetinib will be the MEK inhibitor, so some of that will be able to dictate what the actual endpoints are in the protocol. So the protocol hasn't been finalized yet, but they're hoping that that will be started by the end of the year.
Evan, we gotta keep moving.
We got to keep moving. All right, so the last slide really is, I don't really have any pictures here, but the most important thing is outcomes. Is an area of research that I didn't appreciate and wouldn't have thought of. But there are a lot of posters and a lot of people talking about outcomes, and what does that mean? Does growth equal progression? Does growth without any symptoms equal progression? If you see a little growth, you're going to just start a clinical trial growth. If a child has no growth, but all of a sudden their visual acuity goes down, is that progression? So those types of things are really starting to think about, which is, in my opinion, also an area that is going to really impact our kids. The decision about, that we all agonize over, about when are we going to start treatment again, or what types of treatments, all these types of things, even from cyst control. There was a poster on Avastin being used to control cysts. Some people think that might be useful. So in other words, you may not have to necessarily go for a surgical remediation for cysts. The other thing is the fact that we all know that a lot of our kids do really well 5 years out. But the question is, what about 10 years? And what do we define as overall survival? Our kids are struggling in a lot of different ways. That's not what we would have necessarily defined as overall survival 5 years ago when we started this. So that definition is starting to change as well, because clinicians are recognizing that it's not enough to say the child is alive 5 years later.
Right.
So there's a lot of stuff here that I think just gives me hope that the researchers and clinicians are really going to take a broader look. So again, in the future, I think there's no lack of drugs to try. You saw this list of like 6 MEK inhibitors. The question is which one to use and when to use it. Research is ongoing, but it's not there yet. There's a lot of cool stuff going on, but it's not there yet. And this is a really important point. Amy talked about the fact that we have Dana-Farber as a hub. But the other thing that's really interesting and exciting is because of all the groundbreaking stuff that the foundation has been able to fund, there's interest in the community, in the broader research community, to expand this. And so there's talk of a second PLGA research hub opening in the UK, sponsored by the Brain Tumor Charity. This is huge, because the Brain Tumor Charity is huge, and for them to start to get interested in low-grade glioma research is potentially so that's, I think, all I want to say.
Next slide.
Next slide. So, okay. You're done with me?